signed up as doctors of on call, if you will. They have provided us with their contact information. And so when we come across something where we don't have the expertise on board, we'll pull up our Rolodex of scientists ashore, doctors on call, and we'll give them a call, tell them to tune in, and then they would basically help us guide that dive. And obviously we have these archaeologists in many places. And what we expect to have here that could be best preserved would be most likely Japanese or US flagged ships and planes from World War II. And we have a good network of archaeologists in both of those countries. So we would obviously make sure that we have folks from those places on the line that are the experts of these places.
Can we take a quick look at that little round sponge? Yeah, that one. Just caught my eye. It's very flat. I believe this sponge is a glass sponge in the family Rosellidae.
All right, push in.
Oh, and the substrate that it's on, this is all dead coral rubble from hard sclerotinian corals like the Enelopsamia that we've seen on other dives. So this might give us an indication of what we might see further upslope, a lot of hard corals.
Yeah, and Chris Kelly here, one of our loyal scientists ashore, is a little bit puzzled about this sponge. And if we are set out to do so, we would like to go ahead and collect this specimen.
Okay, let me stop the ship. Bridge nav. Can we hold position? Thank you.
I'm guessing this is very fragile.
This will be a little bit squishy, but it will be fragile.
Okay. Is it a slurp will tear it up?
I think a slurp would tear it. It's pretty small. I wouldn't want it to go through the slurp.
Okay.
Maybe you can put it in the forward bio box if it feels too hard to get around to the starboard.
Yeah, this is going to be floaty, so
yeah,
the forward box probably would be better. I think you're right.
Yeah,
okay.
It's totally open, so go for it.
I'm live at the moment. Let me know if you need me to push in.
Okay. All right, you can push in there.
Yeah, and it'll tell us if it's actually just sitting on top of this rubble or it's actually attached onto a hard substrate underneath.
Yeah, if it's easier to get it with whatever it's attached to, then we can go with that too.
It looks like it's just sitting on top. Oh, nicely done, Jake.
So
It's not over yet.
You just scooped it so pretty.
All right. Retry camera. Or wait a minute.
You can retract camera, yep.
Okay.
Nope, nope,
Oh,
nope.
Oh,
I didn't think it was halted. That's the gotcha with this arm.
yeah. That's one of those features that I don't appreciate about the craft.
I guess it wasn't overly floaty.
Yeah, it's not too floaty because it's got all that rubble attached to it.
Convenient.
But if it didn't have that, it definitely would be floaty.
Is that going to be useful
Okay,
at
you
all
can
to look at?
rack back and
I'm
bump
not
out the box.
sure. It looks
Okay.
pretty deteriorated.
Opening the box.
in here is fine.
Yep. Is this 100?
It is. This is
Wow.
number 100.
100.
Perfect.
Well done. Very nice.
I gotta get this snaggletooth out of here. There we go.
Okay. Closing box. Standing.
Yeah, great job, Jake. Thank you so much, and excellent example of telepresence in action. Our one of our top experts on sponges, Dr. Chris Kelly, on the line, coming across this rare sponge sample that he has never seen before, and that tells you something because he's seen a lot of sponges. Chiming in here, and within a couple seconds we got the video frozen, zoomed in, and then collecting a sample. And not just the telepresence, but also the wonderful
Can I give you a DVL reset?
collaborative endeavor that we're embarking upon. This is all highly collaborative science. Our mission is to explore and
There's a
gain
fish
knowledge
coming up.
about the largely unknown ocean, and that requires a large army of scientists working together.
What is that?
This one looks like a Nizumia. I wonder what Jeff thinks about it. Yeah, that sponge, I wanted to zoom it because it looked different to me, so I'm glad Chris agreed.
Yeah, great eye. Megan, do you have a range to waypoint 2?
Push in there.
Yes, it looks like we've got 250 meters.
Roger that, thank you.
They're fast.
Yeah, they are.
Jake, we have a question for you.
Uh-huh.
So how long did it take for you to get familiar with Hercules, and is it easy to pilot?
It's been several years of coming out and contracting and working with people with lots of experience, like Dan and some of our other pilots. But it really it comes with experience, and kind of similar to driving a car when you're first learning. It takes time, and you get more comfortable with it, and you're familiar with the controls, and little by little, it kind of becomes just like riding a bike, like second nature. Come out here after several months and still kind of just pick it up after a couple dives and jump right back into it. But it definitely took several years to really become comfortable, and it's definitely very situational. So there's lots of different situations that can come about, and it just, experience. All right, come on.
A follow-up question, Jake.
Yeah.
So is Hercules' arm similar to a claw machine?
Somewhat. The claw machines are less functions normally, so usually it's, you're moving in the like a XY plane, you're moving in a direction, and then you just hit deploy on the claw, it goes down and tries to grab the thing. But with this arm, you have 7 functions, and you're controlling it with a miniature version of the other arm, of the arm. So you have a lot of kind of precise and fine movement control.
Yeah, these planars, either one.
Or you can go for a zoom.
Oh, there we go. I wasn't on SPL. I'll do it.
Chris thinks that this is a chrysogorgiid.
Oh, is it? Oh, okay, yeah. I can see that. It's pretty different for a chrysogorgiid too. It's not Rumellagorgia militaris. Would we want to collect this? Yeah, the polyps are very small and they don't have scales. So I think Chris is right. Yeah, this is a chrysogorgiid.
Yep, and we would like to set up to collect a specimen if
Okay.
there are.
Yeah, we're set up.
It tricked me. Bridge, Nav. Can we hold position? Thank you. Yeah, sometimes from far away you get in your mind what something is, and then it's not that thing. But that's what zooms are good for.
How much of the specimen are we looking to collect?
Just a couple branches. If we can get a branching point, that would be ideal.
Can you push in there a little bit?
Yeah, that looks good.
Great. Starboard box, I'm assuming.
Yeah, that would be great.
Okay.
The only thing we have in there is a rock in E, the large outboard
Can
forward.
you come down on the winch 5 meters?
Yep. Just getting a little close there.
Yeah, this should be quick.
Okay.
Just set back down. Yeah, I'm swinging around. Can hit the sample. Nice.
There you go.
Do you think you can get it in one of the little ones?
I think so. Can you open the box?
Opening box. That little fish back there hanging out. Okay, all the way open.
Are they all open?
Yep.
Or yeah, they're all free.
If viewers want to watch Jake with the manipulator that moves the arm, they can go to channel 3.
Alright. Alright, close the box.
Closing box.
Nice. That was the forwardmost one, right?
Yes, forwardmost box.
Great.
Switch back to ROV cams.
Yep. Alright.
Yeah, that was a really strange coral.
Yeah.
Alright,
I've never seen a chrysogorgiid like that.
I'll come up a bit and get
Yep.
back underneath
All right.
Atlanta so we can...
Are you starting to see a few more animals?
It is getting denser.
Yeah.
I heard the last watch saw a Dumbo octopus,
Oh, did they?
Yeah.
No way.
like
Ah.
slightly orange one.
Jelly.
I know.
Well, we need a shark.
That's...
Yeah.
I know they're around here.
What species would it be?
There's a whole bunch of different species of shark. We could see a Pacific sleeper shark or a six-gill shark. Maybe like small lantern shark or catfish kind of deal. Cat shark.
Can we just left of screen? There was something
Yeah.
circular. Oh,
Oh,
well, actually, let's
it's
get the fish
Oreo.
first.
Okay.
Oh, yeah, Oreo. Okay.
Push in on the fish quick.
Yep.
It's got a parasite on it. Looks like an isopod parasite. So isopods, for people who aren't familiar, are like the roly-polies you might get, or pill bugs you might see in your own backyard.
That's a pretty big one.
It is a big one.
Will
All
it
right.
ever fall off?
It could. Maybe if the fish isn't doing well, the isopod might go find a different host. But it seems like
Yep.
it's got a pretty good thing going for it right now.
Is it feeding off of it?
Yes, it is feeding off the fish.
Push in there.
Yep.
I think that's the base of a dead coral. That yellow.
Yep. Yeah, thanks, Jake.
Yep.
Is it-
Okay.
The fish isn't ready to be done.
No.
Yeah. It's like, oh, check out all of my good sides.
Yeah, I'm giving you all the angles.
Oh, as I run into the rock.
Coming wide. What exactly is it feeding off of on the fish? Is it dead scales or?
They usually feed off blood.
Oh, okay. Fair.
Quick, that urchin, sorry.
Sure. Yeah, that's all right.
Yeah, so that's a econothurid urchin in the subfamily Spheronematid.
Push in there.
Excellent, yeah.
Okay.
I'll try to get a good zoom of every one that we'll see a new one, just so we have that
Oh,
documented.
and there's a little polychelid blind lobster. There's someone.
Oh, right there. It's tiny.
It is. It's really tiny. Usually they're bigger,
Push in.
but they have to start small at some point.
Oh, so tired.
Oh, yeah.
I just
So this is called Homerian. It is a blind deep sea lobster.
Come on.
They're not related to lobsters that you eat. They're a sort of ancient form of crustacean.
How's the focus, Daniel, on the cinema cam? Is it better?
Well, Makani, what are your thoughts?
Kinda.
It's not that great.
Okay.
Yeah,
That's unfortunate.
yeah.
I haven't had a chance to play with it yet.
But maybe, since we're already kind of well established here, we can run around with a quick rounds of introductions here in the control pan.
So, Ali, Milsuelvo, good afternoon. My name is Imada, and I am in the SCF seat, or Science Communication Fellow seat.
Alihi, good afternoon. Aloha, everyone. Daniel Wagner sitting in the watch lead seat and also serving as expedition leader for this expedition.
Ali, Milsuelvo. My name is Maikane Andres, and I am the scientist for this watch.
Hello, everyone. My name is May Lubetkin, and I am the data logger on this watch and the science manager on this cruise.
In the front row, my name is Megan Putz, and I am in the navigator seat.
And my name is Jake Bonney. I'm driving the bus. Hercules. Stole that line from Dan. Yeah. Sometimes it feels like that.
my name's Sarah Surgeon, and I'm in the Atalanta seat today.
And my name's Erin Rainey, and I'm in the video engineer seat.
The bus.
It is yellow.
Yep. And we're all in it.
And then back in the peanut gallery, I'm Jeremy Wyrick. I'm director of NOAA's Office of Ocean Exploration, just sitting in, listening.
There's no camera for me to get on you.
Didn't know you were back there.
This is exactly where I need to be, off
Oh,
camera.
wait, no, you're a little bit in camera there on my bottom right.
Then I gotta scoot back.
Push in there.
Yep.
Oh, here we've got a small sea urchin. This one is called Aspidodiadema. Its notable characteristic is these super long spines.
Yeah, my Connie had spotted this from way back out on the cinema cam.
Yeah, good spot. I can frame up a cinema cam shot.
Okay.
Oh, did you see all those super, super tiny ophiuroids on the rock?
You want to try one?
Yeah, just zoom all the way in, and there's like a whole bunch of them. They're really little.
Do you want me to zoom back in with this camera?
Yes, please.
Yep.
You see them now? Look how small they are.
They're tiny.
That was cool. Thank you.
All right, come on.
Yep.
Wonderful, thanks.
We can't let Dan's watch get the best still, so we have to work together.
Well, who's judging the still contest?
I think it
I
might be
will.
rigged because Dan is.
I think the video engineer teams should,
Yeah, that should be it.
because we'll be non-partial, non-biased. Sort of.
Sort of.
But then at least you have one person from each watch.
Yep.
Yeah, let's try a real snap zoom on.
Yeah, I think that's that same chrysogorgiid we just collected. You know? It's got that feel.
Yeah, it does.
But from far away, it's definitely like given more primnoid than
Push
than
in there.
chrysogorgiid, which is what caught me. But it definitely is a chrysogorgiid. This one has its polyps all retracted, so they look like little snowballs. So cute.
That's great.
Yeah, the notable characteristic of chrysogorgiid polyps is that they tend to be very small. They don't have much of a body, and the branches are very, very fine.
All right, come on.
Okie dokie.
I'm excited to find out what that one is, if it's been described before or if it's possibly new.
The sample we just collected?
Mm-hmm.
I'm personally very, very excited and anticipating eagerly what we're going to see as soon as we make our sharp 90 degrees turn to.
I have a feeling there might be a bunch of sclerotinian reef structure up top, just given how much rubble we're seeing down here. I've seen this before in the Northwest Hawaiian Islands and Emperor Seamounts. Right there at the end of the chain, there's been findings of these large sclerotinian reef structures at about 700 meters, and that's sort of where we're reaching to in depth. So that's what I expect to see. See, there's some more of that rosellid sponge, just larger. Bridge nav. Can we make a 40-meter move, 145? Thank you.
Starting to see some larger glass sponges, polyopagons,
And I think this other big sponge over here might be similar to the one we collected, just a larger version of it. There's a metallogorgia with its associate snake star, Ophiocreus oedipus. Lots of big polyopogon.
Back out ahead. Oh, you already started to move. Nice. One step ahead of me.
Yep.
Per usual.
Yeah, once you start getting ahead, then I call in a move.
and I think as soon as we reach the top of the ridge, I think we'll initiate another automated stills sequence. Still have a few hundred meters to go. Here's another one of these hermit crabs.
One's got a real shell, I think.
Yeah. So all these yellow corals are Acanthogorgia. They're very sort of fluffy looking, and their polyps don't fully retract if disturbed. Further back, we have a primnoid coral, the white one. And then the sponge is a Poecilastra.
Yeah, maybe let's get a quick zoom on this heterocarpus, I believe.
Yep.
All right, Aaron, you can push in.
Oh, you can see all of its mouth parts.
Huh.
That's great.
Look how flexible that arm is.
Is it scratching itself?
It's probably cleaning itself. So shrimps have 10 legs and then 3 sets of mouth parts.
All right, we can come wide. Gotta keep on moving. Ship's still moving.
Yep. Yeah, but this is all heavily encrusted carbonate. Early in the dive, they did collect a rock, and that was carbonate. And you can see here in a few places the carbonate being exposed, that white limestone. An indication that is all formed by corals, hard corals, crustus colorine algae, and a whole bunch of other organisms that secrete limestone and then cements together and builds these massive structures. And here we are at a depth of close to 900 meters, 870 meters right now. So these places, the substrate here was not produced in place. This was all a lot shallower at some point in the past and has since subsided. And now one thing that's interesting about this dive site is it's a pretty lengthy ridge feature, so pretty narrow. And that ridge goes, we're currently just on the periphery of the Palau National Marine Sanctuary, about 5 nautical miles from the boundary, and that ridge feature actually transcends.
Yeah, that over to the right, there's a benthopectinid sea star.
Sorry to interrupt.
No, good eye, please do. Well done, Aaron. Well spotted.
All right, you can push in there.
So this is a sea star in the family Benthopectinidae. Probably benthopectin.
Do they have a common name?
Not beyond sea star, I don't think. A lot of the deep sea species don't have like specific common names. But some of the notable characteristics is this like really beautiful red pink color with the tips of the legs being slightly lighter in color, and also the very distinctive spines around the outsides of the arms.
Yeah, you can come forward. Thanks.
There's also a black coral up to the left there.
Yeah.
Could be Trisopathes.
You want to zoom on it?
Yeah, let's get it just for good measure.
Okay.
I think Megan is right.
On a rock. Quick zoom there.
And then there's also a little Europtychus squat lobster in the branches. And there is also a snake star.
That's great. Thanks,
Okay.
Jake. So as I was mentioning, the feature that we're diving on is it's a much larger ridge that basically extends across the boundaries of the Palau National Marine Sanctuary, and part of the mission here is obviously trying to explore and characterize and map what's in the Palau National Marine Sanctuary. And yeah, beautiful big boulder coming up here right in front of us. But not just understanding what's in the sanctuary, but also how that is connected to those ecosystems and habitats outside of it. And the connectivity between these habitats is a really important feature to understand. Many of these large marine conservation areas have spillover effects, and so understanding how these
really
offshore
tall.
places replenish stocks inshores is an important thing to understand. Looks like we have those larger gooseneck barnacles there on top as well. And those yellow crinoids. Yeah, very similar to a lot of species we saw yesterday when we're about 50 nautical miles south of here. And the other unique thing about today's dive is that this ridge is actually pretty close to trench habitats, and just close to us, about 20 to 30 miles east of us, Jamstec did a series of dives over the year, most recently in 2013, but also in the 90s, exploring depths of 6,000 meters and deeper with their human-occupied submersible Shinkai. And so we on purpose are basically also picking sites that are somewhat adjacent but much shallower, and so that we're trying to understand vertical zonation patterns and how these very deep ecosystems might connect to intermediate ecosystems, and then also might be connected to very shallow ecosystems. You know, Palau is very unique in that you have basically the trench and some of the deepest ecosystems on the planet being very, very close to the islands. And that creates a very interesting connectivity of ecosystems. And in particular here, because we have pretty persistent currents that connect this part of the Western Pacific to the east. So we have two major currents, the North Equatorial Countercurrent and the North Equatorial Current, which flow east and westwards respectively. And here on the eastern part of the islands, basically intersecting directly with the islands, and then on the western side of the islands, creating some large circular ephemeral circulation features known as eddies. So these are several kilometers in size. They will rotate around and persist for weeks to months at a time. And these are also places where you have higher aggregations of fishes.
Is it hatchetfish?
Could be.
It's swimming like one.
Whoop.
It's shiny like one.
And it's gone.
It was probably a hatchetfish.
Is that another one of those large chrysogorgiids?
Yeah, it is. It's the same thing.
Perfect. Yeah, that's great.
We didn't see that one on the other dive.
Yeah.
It's very interesting.
There's a fish coming up to the left side.
Yeah, it's great. My Connie has got the cinema cam display and can see things ahead of us.
Push in there.
Oh, yeah, this is a tiny rat tail fish. I think this is like the one that we tried to catch yesterday. I know Jeff would be happy if we were able to catch it, but they are very, very fast.
Yeah, let's see what he says. Jeff thinks it's probably Kumba.
Want a better look at it? Or
Yeah, I think we're okay.
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 The previous watch collected 2 fish already.
Oh, they did.
So we might, I don't know if this is different. Jeff is typing, but
Yeah, I didn't know we had fish collections already.
yeah, the first 2 slurps are both fish.
Sweet.
Yep. One is a small black rat tail and a bathy gayid fish.
Very nice. Well, as we get shallower, we might have more chances to run into the coelurhynchus we're looking for. So coelurhynchus are in the same family as these rat tail fish that we're looking at now. They just have a very distinctive shovel-shaped head. Ooh, that urchin looks cool.
That's really cool.
Yeah, let's take a peek.
So this is a pencil urchin, urchin in the family Sideridae. This looks like it could be Histosideris.
I have more push in if you want it.
Okay, yeah. Go for it.
That's it. I thought I had more. Wow.
Beautiful.
Yeah, gorgeous.
Yeah, it's spectacular.
That's really nice.
So are the
So interesting.
like small red, they almost look like spines. Are those soft and then the white ones are hard? How?
So yeah, the little spines around the base of the long spines are hard as well, but they have tube feet in between, just like other urchins and sea stars.
Wow.
But the tube feet are very fine and small.
Yeah, that's great.
All right.
Stunning.
Come on.
I have a feeling there's going to be quite a few screensavers made from the imagery collected on this expedition.
Oh, absolutely.
Maybe that's what Nautilus should offer. The Nautilus screensaver collection.
We could do a calendar.
We should totally do that.
That would be December, I think.
Yeah,
I think
Yeah,
that
that-
would be December.
it feels very December.
Here we're coming up on these large gooseneck barnacles that are about to come and screen. Another one of these
Let's check out that fuzzy white one.
Fuzzy white one.
Yeah,
Is this one? You're talking
yeah,
about?
that's the one. Is that the same thing? It looks really fuzzy. There's also a crab on there holding a little piece of primnote or plexaurid or acanthogorgia coral.
You can push in there.
See, this is what I thought the other thing was, the chrysogorgiid that we collected. This is a Narella, which is a primnoid coral. And it's got some zoanthids on it. But when we saw the other one, didn't it look like the same thing from afar?
It did, yes, indeed.
So, yeah, that's how it stumped me. Because I've seen this coral before, but that chrysogorgiid one I hadn't seen. And it looks like this homolid crab, the carrier crab's carrying a nice little piece of acanthogorgia in its legs.
Nice.
Yeah, it's kind of like a little flower in your ear.
Mm-hmm.
Like a lay.
So fashionable. They carefully curate what they carry.
Barnacles over here.
Oh yeah, those are awesome. That's a great zoom of that. I'll have to do some research to learn a little bit more about gooseneck barnacles, but I've never seen these really big necky ones that cluster like this. So those are a bit new to me. We don't have them in Hawaii.
All right.
That's
Yep.
wonderful.
Well, now I know for annotating that not only do we have chrysogorgia corals that are white and fluffy, we also have the Narella that is white and fluffy and looks almost exactly the same from far away. So we'll have to be very careful when annotating and not make assumptions.
Oh, I see a little fish.
Yeah, there's a few big.
Good eye.
Well, that's a really little fast one. Okay, I think this is a bristlemouth fish.
Or oh, it's getting away. I was going to say, are we going to do a snap zoom?
It might have gotten tired, and you might be able to zoom it now.
I lost it. Where'd it go?
It's below the lasers.
There it
Like,
is.
there it is.
All right, you can push in there.
Okay, I'm going to go quickly. Sorry,
Yep.
everyone.
No, to do it. Yeah.
So this is called the cyclothone. It is the most abundant vertebrate on the planet.
Whoa.
Wow.
Wow.
It's getting away.
It's doing a very good job.
All right, come on.
Yeah, it's hard to get a good picture of those. They are a midwater fish, so you find them not usually this close to the bottom, but we do often see them on seamounts because the seamount comes up off the seafloor quite a ways, so it's part of the water column in many ways.
And I assume these are vertical migrators?
Yes, yes, they are.
Yeah, and that's also the exciting thing of this particular feature is that we're intersecting the depths where you typically have strong vertical migrations, 1,000 to 200 meters. And so yeah, this feature would directly intersect that. The top of this feature is about 700 meters or so. Yeah, looks like one of those big, what we've been calling Poliopogon, but Chris mentioned that it's been changed to a different group.
What's this? A sponge?
Yeah, that one looks a little different.
Push in there.
Looks like it's got little legs.
It's a sponge?
It's a sponge, yes. This looks like a glass sponge. It's kind of a strange looking one. I wonder what Chris thinks about it. Has a very regular dictyonal framework. That's the sort of cross-hatch pattern of spicules that we're seeing.
Does he need to see it more?
Yeah, can we like maybe focus on those little tubes up top? Because that's the strange part.
This is the furthest I can
Yeah,
go.
okay, yeah.
The closest I can.
So, okay, that's good.
Okay.
Yep.
Come on.
Chris is suggesting Holoplacidae.
Okay.
Another big Metallogorgia. Try, yeah, let's take a quick zoom here.
All right, push in there.
Looks like a paragorgia. And there is a snake star on it.
And there are species within this genus of the bubblegum corals that can get massive. I've seen the specimen that's like the size of this control room almost. Like they're enormous.
That's up in the northern.
In New Zealand, yeah, the trawls.
Oh yeah, like very cold water.
Yeah.
Shallow cold water with lots of food.
Come on.
Yep.
The base is so wide on this one. Is that pretty normal for a fan of that size?
It's definitely advantageous to that coral to have a wide base. It'll be less likely to be knocked over.
Catching that fish.
I'm gonna go chase this fish.
Is that kind of like primary growth with trees? Like, would a coral want to first put all of its energy into making a
Push in there.
strong base before
I would think so, yeah.
fanning out?
Here we're looking at a eel in the family Synaphobranchidae. This one. We're looking at the dorsal fin origin and the anal fin origin to see which one starts first. This looks like this is in the genus Synaphobranchus, and the origins are very similar. So it could be one of two species, Synaphobranchus affinis or Synaphobranchus calpii.
All right, come on.
And it looks like we're getting closer and closer here to the top of the ridge. So we'll try to make it up to this ridge and then take our 90-degree turn, heading northeast, and then we'll follow along the ridge. We'll also at that time activate our automated stills to take a series of photographs along the ridge feature.
Can we also take a Niskin while we're on top of the ridge?
Yep, absolutely.
Thanks.
Looks like we're coming up on large wire corals. Probably bamboos.
Oh, this little pink fuzzy one here at the bottom. That looks different. Oh, it's a soft coral.
Catch a little fish in the background.
Have we seen any soft corals besides Anthomastus?
I not on this watch.
No, I don't think so.
You can push in there.
Yep.
Yeah, good eye, Megan.
Yeah, so this is a soft coral in the family Nidalidae. Could be something like Dracemia.
Oh.
I also spot a small sclerotinian up in the right, that yellow polyp.
There's a big fish coming right across.
Oh,
All right.
yeah,
Come on.
that's a Synaphobranchus.
Same one we just saw, or?
Yeah. And if it's too hard to get, there's also requests on these wire corals. There's two. There's one
Okay.
directly left or one directly in front of us.
Now, these ones have some really dense polyps. Like really, really dense. Yeah, if we can
Push
get
in
a
there.
close up of the polyps. Is this a primnoid?
Yeah, I think so.
Ah, fascinating.
There we go.
And there's a Narella, I believe, that has that kind of.
I don't think it's a Narella.
And a Candidella, right?
I don't know. I feel like we should collect this. This is
bit.
different.
Let me
Yeah,
get this.
Chris thinks
Come
it's
on
a
for a second.
Narella. Yeah,
I've never seen a Narella this tall before.
Yeah, if we're set up, Jake would like to sample this.
Do you want a better image of it before? It was a bit too close to...
Yeah.
close
Sure,
focus.
yeah, that'd be good.
Here, you can push in there.
Thank you.
Push around a bit. Oh my god.
Yeah, this could be something new. These unbranched Narellas aren't well known. And we are at a really interesting depth, so
Sorry.
between 800 and 1,000 meters isn't super well studied.
Why is that, Megan?
Well, we usually seem to target a lot deeper areas when
Alright.
we do our dives.
Come on.
And the tops of a lot of seamounts tend to be deeper than this. This is a relatively shallow seamount. But we just also haven't done enough exploring either. So this area could be very exciting. And we have a lot of opportunity to find new and exciting things.
Indeed.
Yeah, we can get a little haircut and slurp it.
Yeah. Could probably get a better zoom now that I'm stable.
Okay, yeah.
If you want
Do
to
you
push.
mind?
Yeah, let's go for it. Yeah, the polyp shots will be very helpful to the scientists that go to describe this. Seeing a specimen in the lab is very different than seeing it alive.
Definitely.
And it can be very informative to know what it looked like in its natural environment.
That's as close as I can get.
Okay.
So yeah, Chris thinks likely an unbranched Narella. Unknown species. Thanks, Chris.
I'm curious to find out how many polyps are in each whorl. So It looks like all the polyps are in like tiers, and they're just so densely packed. There seems like a lot of them. We usually don't see it that dense, which is why we think originally thought it was a bamboo, because it looks and acts very much like a bamboo coral. This is a dive of tricky corals where they look like other corals, and we feel very confident about what they are, but they're not. Just goes to show we still have a lot to learn.
Okay. How much of a piece do we want?
Like maybe
This.
6 inches.
Just get my tape measure out.
Yeah. You know, 2
Oh, I got
claws worth.
Still have a piece of that other coral that was
Oh.
How's about there?
That looks great.
There we go. Starboard box. You can switch over.
Yep.
You can bump open the box.
And all the way up. I like its little, like, poofy cap.
All right, it's in the
Sweet.
box B.
Awesome.
All right, close the box.
Thank you.
Well done, Jake.
Yeah, outstanding collection.
Sorry, Jake. We'll hold off on the Niskin until we get to the very top of the seamount.
Okay, no problem. Just let me know when.
Thanks.
Switching back.
All right, that's great. So just as a time check, we have about 10 hours, 10 hours of bottom time remaining. We'll have to cover about 1.4 kilometers if we want to make it all the way to our waypoint, our final waypoint. And that should be pretty interesting. In fact, we're going to be approaching the top of the ridge here, about 50 meters or so, and then start moving in a different direction, so along the ridge. And then when you look at the fine scale of this ridge, it's narrow, but it has several topographical highs. There's 3 distinct peaks among them. And so we'll try to reach at least 2 of those, and there's actually one that's past what we have set out to do, and that's the very, very top of this feature at less than about 600 meters or so. So we'll see how far up this feature we can make it with the 10 hours we have remaining. And then, as I mentioned, after we pull the vehicles, we'll have to be pretty much on time because we have about a 6-hour run from here to Malakal Harbor. We'll be pulling into port first thing tomorrow morning. We'll be alongside there over the weekend before departing again on Monday morning for another 11-day expedition. And that will exit through the West Passage, and we'll be working off the west side for the first 4 days, exploring various different features, and then spending the last week or so working around the southwest islands. And that should be a very interesting. Those are thought to biogeographically be somewhat distinct than the other places around the Palawan Islands and ocean ecosystems. And I'm particularly excited what we'll be able to tell after this is all said and done. We will have completed a complete 360-degree survey around the ocean of Palau, surveying starting in the north and in the east and the south, and then finish and then the west. And so that will be a good representation of some of the diverse habitats that have never been explored in Palau. And we'll also have covered a wide depth range. We'll include on this next expedition dives probably approaching 4,000 meters, a couple of those. On this expedition, we did a couple to 3,000 meters.
Let's zoom that sponge, the white one coming in the middle.
There's another big sea star up there.
Mm-hmm.
Push in there?
Yep.
I feel like I've seen this one before. Oh, it's not in that gallery.
Yeah, so Chris is Aloplax.
Aloplax, yeah.
A pheronematid glass sponge that was recently described in the last several years. Thanks, Chris.
If you come wide, there's a fish in view. Should be. Might be up a little bit. To the left. There it is. As far as I got.
Oh, that one's a different rat tail fish we haven't seen yet.
Well, let's go look at it.
It's coming to look at you.
Yeah.
Indeed.
I believe this might be a Godomus. Has these really long filaments on the fins.
We have one speculation here of a bathygatid.
All right, push in.
Oh, that's a great look. It's a star.
Doesn't look too happy.
Oh yeah, this is a morid. See the long filament?
It's gonna hit the camera.
It's gonna get too close.
Oh.
I can't focus.
Yeah, so bathygatus.
That's the
Thanks,
near field.
Jeff.
Wow.
It's like, I'm gonna explore you.
Do we know how long this is?
It's probably between 30 and 50 centimeters.
All right.
Yeah, I think that's great footage. I think maybe in there.
Yep.
There's another one of those sea stars with the white tips. You just went
Mhm.
over it. What was it called?
Benthopactinidae.
Benthopactinidae.
Is this a,
I'll get there, Megan.
looks like two large black corals here.
Well, I think the one in the background is that chrysogorgiid.
Oh, yeah. Yeah, right.
It's tricky. It definitely doesn't look like a chrysogorgiid.
Can zoom in on this fern-like black coral right in front of us.
Yep. Push in there.
And then we got a sternostylus squat lobster.
Quite the entrance.
Mm-hmm. Oh. There's also a lot of little amphipods crawling around.
Yeah. Well, yeah. Beautiful in view there. There's a paper written in the early 2000s of a black coral colony collected off California by Milton Love and colleagues, and they had just collected one big fan and then basically dissected out of there every single invertebrate, and they counted on the order of several thousands of tiny animals that were found within that coral. And yeah, there's obviously some big things, but once you start looking at the microscopic level, these things are really a massive habitats. They're the skyscrapers of the deep.
And you wanted to look at the other coral behind it too? Just.
What do you think, Megan? Is that distinctive enough? Yeah.
This coral?
Yeah, the
Come out a little bit.
chrysogorgiid that we think behind it.
Oh yeah, I think it is the same chrysogorgiid behind it. We can zoom in real quick and confirm.
Yeah, push in there.
Yep.
Yeah, it definitely is.
Wonderful, thanks.
Okay.
Yep.
So, what genus is this black coral in?
This is Bathypathes. And so these are
It's very like densely branched for Bathypathes.
Yeah, incredibly dense. But I have seen a few like that. Maybe a quick zoom like there.
Yeah, those are a grouping of soft corals. Oh, and there's a crab as well.
King crab, I think.
Oh, cool.
Yeah, there's a bunch of really fun, unusual stuff on this dive.
Push in there.
I'm gonna look at the soft coral gallery.
Oh.
What's that little thing that's in between the coral?
I think that's another one. It's just all closed up. And
Okay.
yeah, sometimes they do that. They'll just pull everything in, and since they're soft corals, they can close down really tight. So these soft corals in their family Nidalidae could be something like Gersemia or something like Dendronephthyla. Dendronephthyla are two potential IDs for this one.
Too close there.
Hopefully we can get an expert on the line who knows these better, might have a better ID. I'd also really like to take a look at that crab.
Okay. And combine them.
Oh yeah.
Bump on over to the crab.
So this is a little king crab. Oh, it's
no.
fast. Oh no. It's in the family Lithotidae.
You can push in there. Spiky.
Very spiky. So this could be Neolithotes longispina. That's great, thank you.
Okay.
Yep.
Hello, this is Harold in the lounge.
Yeah.
Hi, Harold.
That crab looks pretty similar to something we saw quite a bit on the baited cameras, and Jeff is interested in collecting it if possible.
Well, that sounds tricky.
Yeah.
It is.
Yeah, that's
Scoop is Jeff's suggestion.
I feel like we should suction it
Well,
so it
whatever
can't run away.
whatever you think is
Otherwise
best.
it's gonna run away.
I won't be able to scoop it in underneath that rock either, so
Yeah.
suction's really the only option. You could try it.
I think it's large enough that we can just like pick it up with the suction,
Yeah.
but it's not gonna,
on its back or something.
yeah, on its back. It's not gonna go through.
Yeah, I mean, we can give it a try, but if it gets
Yeah,
too
and
tricky,
if we
we'll
put it
avoid.
in the
Yeah,
forward box and then we can't open the box.
It won't eat anything we have
Yeah,
in the box,
that's-
will it?
Do we have anything in the box?
Yeah, we do.
With
We
a
have,
sponge, it won't eat a
yeah.
sponge.
Okay.
It doesn't like glass shards?
But does that negate us then from
Nutritious
collecting other things?
Sorry?
Does that negate us then, or limit us? And
Is the scoop still in the forward omega?
Yeah.
I mean, have we used the scoop much?
I haven't touched it.
No.
All cruise. I don't know about other watches.
I feel
but-
like we've kind of lost that sort of omega box to the scoop, so we could also just put it in the scoop box
perfect, yeah,
since we haven't been using it. Yeah.
yeah.
And then
okay.
that keeps it away from whatever's with the sponge if there happens
Yeah.
to be other organisms,
Yeah.
associates. I think I saw a brittle star associate with that sponge.
Would it eat a brittle star?
Probably not.
Okay, so I guess either forward box would be okay.
Yeah, it would probably try to catch any small amphipods that might have been on the sponge.
Just wasn't sure if we're putting it in a box full of food.
It's like, here, have some free snacks
For now we can
and a bus ride.
Enjoy the ride.
keep it low. Yeah. can push in a bit.
Yep.
You can maybe increase suction.
Oh, it's going
I
away.
think it ran away. Can't see anything.
It's right there.
Yeah, I think the
Yeah.
spines are working against us here.
All right. Yeah, that's gonna be tough.
Making an escape.
You might have to try to get him from behind.
These can get really big, can't they?
Yeah, they can get pretty big. Some species get larger than others.
It's about as far as our reach I got. It's fast. It's a fast crab.
Well, until now.
Oh.
Yeah. It's too, I think it's gonna be too fast.
Yeah, I think if we
And
well,
it,
just
if
move on.
anything, the spikes are preventing the suction from...
Yeah. Yeah, thanks for giving it a shot.
Yep.
We'll continue on.
That was a challenging one.
Jeff says thanks for trying.
Yeah. It's fun to try, that's for sure.
Yeah, maybe one of the smoother ones would be easier.
Yeah.
Sometimes these animals know how to stay away from ROVs pretty well, so
Yeah, I
they're
think
meant to stay down here maybe.
their spikes have also reduced a predation event.
Doing what they were made to do.
Mm-hmm. Works on all kinds of predators.
purposely designed to avoid ROVs. He knew this day would come.
Exactly.
All right.
All right, looks like they're getting very close to.
Yeah, one more move. Bridge, nav.
Yeah,
No shark yet.
No shark, but lots of different kinds of fish.
That's true.
Yeah, this one looks a little different.
Push in there.
Yep.
What is it, Jake?
Cosgill?
Yeah.
Just a really small one, right?
Yeah, this one kind of looks like Barathierides. Hopefully Jeff will chime in and correct me if I'm wrong.
Jeff wants to collect it.
All right.
Okay.
He's not sure.
You come wide?
Yep.
Bridge, nav.
That would be fish number 3 for this dive.
Can we hold position? Thanks.
Okay.
Thank you.
All right, suction all the way up.
Oh.
So close.
I think we spooked it.
Yeah. That was pretty scary.
I think we can keep moving. Jeff says this one is a really strong swimmer, so.
Yep.
Yep.
Stronger than the suction.
Mm-hmm. That's Oreo. All right.
What do you think about that Niskin? We're getting pretty close. Like, honey.
Bridge, Nev.
I think we'll take it when we get to the
Can we make a
700,
30-meter move,
Okay.
135?
Got it.
because that's when we start
Thank
doing
you.
our ascent.
Yep.
This is a basket star in the family Gorgonocephalidae. I love how they move all their arm segments and curl them and unfurl them. What type of coral was it sitting on? It looks like that chrysogorgiid.
Do you want to look at it, or
Well, did anybody notice what type of coral it was sitting on?
I didn't? No.
No, I didn't see.
Oh, I think that's our friend. We can get a good look at it, but there's no way we're suctioning that.
You can push in there. This one looks a bit bigger.
Oh, this is a different one, yeah. Look at those really large pectoral fins. It's really cute. What is it, Jake?
Cuskeel. Alright. Coming out.
So when do we find out who got the best still image? Because I am out to win this thing. I need to know before I depart the ship.
I love it. Good motivation.
Also the perfect amount of confidence. What's that? Oh, it's an Oreo.
Monometopus. Maybe the other little one that we were looking at before was Monometopus. Now I'm looking at all my cuskills because I gotta know.
We're about to come up on a very large colony.
Daniel, did you say that we're starting photogrammetry at waypoint 2?
Yeah, once we're there, I'll
Okay, nice.
just give it a, see how we do.
Sweet. I think we're just about there, right, Megan?
Yep, we're just about there.
Cool. So,
Maybe as soon as we turn our heading and head northeast, I'll just initiate it then.
sounds good.
And we'll try until at least waypoint 3 and see how we do. But yeah, I think this is where we'll start seeing the action.
Nice.
And it'll give, I wonder if it's going to increase or decrease Makani's chances of getting her prize.
It's hard to make those adjustments every 2 seconds.
All right, so we initiate.
Yeah, we're pretty much right on the money.
All right. Kick us off, Makani.
Do we want to do a Niskin here?
No, I think we'll do it when we, right before we start ascending at the very top.
Oh, at the very top.
Yeah.
Yeah, not here, Jake.
I'm moving the arm
Okay.
out of the, it was in the upper camera.
There's our first scleractinian. Yeah, that's alive. At least that I've seen on this watch. And we can see what was Megan was alluding to it earlier, a lot of rubble from these
It's going to
hermetid.
get much shallower, I think up to 600 meters, and our distance to the top at waypoint 4 is about 730 meters.
Can we take a quick look at these guys?
Sure thing. All right, push in there.
I think this is a dead Metallogorgia. It's been recolonized by a bunch of hydroids.
Yeah, I think you're right. Yep. And you can see the golden coloration of the skeleton there. That's also one of its common names, although there's several things that have the common name gold coral, including some colonial zoanthids. That's great. Thanks, Jake.
Okay. Oh.
Oh, it's very flexible.
The arm's sticking out a bit.
Okay, our new heading is going to be 068.
Yeah, and right here up on Q, as we're moving across the ridge, we'll see higher aggregations of animals almost instantly. And we see also starting to see some bigger colonies. And again, we see all the sea fans all oriented in the same orientation. And those are nature's current meters. They give you an idea of what the currents are doing here, where the currents are flowing from. Everything here is positioned in its perfect orientation, so it's intersecting the predominant current flows. And we are pointing to the northeast right now, bearing 160, and we see they're all aligned with us, so
Another crab.
currents basically flowing parallel to the ridge here.
Squat lobster.
Squat lobster.
Yeah, this is again a good illustration of what Megan was talking about earlier. All that rubble, that's all material made by hermetipic deepwater corals. A couple different species we have seen on this expedition are in the genera Madrepora and NA167-100. There's in shallow waters we have several hundreds of reef building corals. In the deep sea we only have about half a
There's
dozen.
a sea spider
Pycnogonid.
on the white coral.
Push in there.
This is in the giant sea spiders.
So this is in the family Colacenidae, and it's called Colacenidice. Yes, it eats coral. So it has that long proboscis structure, and it will suck out the insides of coral and anemones.
That's great.
All right. Alrighty.
And then there was also a pink something on one of the corals in the lower left after we zoom out. Yeah, just on this rock.
That shrimp.
I see the little pink.
Oh yeah.
And we gotta,
That be a
yeah,
predating
sea star
sea
eating.
star.
It could be. It looks like that's a distinct possibility.
Push in there.
Let's see. Nope, not a sea star. This is a mushroom coral.
Huh.
Wonderful.
Cool. All the polyps are retracted right now, so it's just a little pink bulb.
Okay.
All right.
So for those who are following along with the dive plan, we have now reached waypoint 2, about 720 meters from where we started to dive about 6 hours ago. And now we have about a kilometer and a half to go, moving along the ridge. And there's one of those sponges we saw yesterday on the right of the screen. They were very common yesterday. And let's see if they...
Let's check out that bamboo. So this is
That's
an
still sharp.
internodally branching bamboo coral, meaning that the area where the coral branches is between those black nodes.
Nice. Do you want to zoom on it?
Sure.
All right, push in.
Yeah, the associates are some basket stars.
Is that a crinoid?
Yeah, and a Crotelometra crinoid. Those are very distinctive because they have those dark black feathers with the yellow cirri.
Ah.
Oh, and then there's a Sathometra crinoid, a different species of crinoid.
No
as
yellow
well.
tentacles?
No,
on that
yeah,
one?
no yellow cirri. It has really long pinnules at the ends of its feathers, feather arms. That's great, thank you. Bridge, Nav. Can we make a 40-meter move 068?
068. Roger.
Thank you.
Looks like another Oreo potentially. A lot of acanthogorgiids.
I was gonna say.
And yeah, we're starting to see the little stubs of a lot of these hermatypic reef building corals. A lot of Nalipsami, all those white little harder pieces. And they are growing on some coral rubble.
Hey Jake.
Yeah.
Just wondering, what is the zoom set to for the upper cam? Is it -7 or 9? We're not able to see that back here, so
1 second.
thanks.
Upper, you want -7?
We
upper?
want -6, but I heard apparently that's not possible.
I just got to -6, but I don't know, the vehicle's in the frame. Is that okay?
Yeah,
Is that what they want?
that was the instructions we were given, but
Okay.
it does seem a bit strange. What was it said before? Do you remember?
I think it was zoomed all the way in past the.
Do you remember the number?
It was like all the way, whatever.
Okay.
Yeah.
Is the current starting to pick up?
Yeah.
Is that-
From left to right across the Hercules screen.
Yep.
So north to south.
Nice little colorful assemblage here, and I think my Connie is going to be starting to win some competition here. This is
Wow.
quite stunning.
Yeah.
Prize is all yours.
I need to ask Dan to make me a special prize.
A special keychain?
Yeah.
Good job on the focus. Looks good.
Yeah, that's.
Do you think it is better? Yeah.
It's quite lovely.
Do you think we could turn the lasers off for just
Sure.
a moment to get some nice laser-free video? It is true that it catches itself, the autofocus. But yeah, when it goes out, definitely was not doing that yesterday. So that was just cleaning up oxidized cables or something?
That's what Nan said.
Yeah, it's good catch.
So if you are just now tuning in on nautiluslive.org, we are currently exploring an unnamed ridge southeast of Angar, approximately 30 nautical miles. And the ROV Hercules is currently at a depth of 810 meters.
Yeah, thanks, Imada. And if you just joined us, don't worry, there's a lot more exploring to go. About 9.5 hours to be more precise. That's as much time as we have to explore, so we'll try to cover some ground here and make it to the top. There's several, so we are now reached the top of this ridge, and there's several kind of distinctive peaks as we'll move parallel to it. We expect to see a lot of animals here at top. And then this will be the last of Lebu's Voyage 1 expedition, the first of 2 expeditions aboard exploration vessel Nautilus focused on mapping and exploring the Palau National Marine Sanctuary. These are the first of several contributions sponsored by NOAA's Office of Ocean Exploration and Research through the Ocean Exploration Cooperative Institute in support of the U.S. government commitment to support mapping the Palau National Marine Sanctuary, a commitment made at the Our Oceans Conference in 2022. Conference co-hosted by Palau and the U.S. government to address cross themes on climate change and other topics, and showcasing the long history of collaboration between the U.S. and Palau. And it's been truly exciting to be part of this. And so we'll have a lot more exploring the Palau National Marine Sanctuary, another expedition right after, and also next year.
There's an orange, I think it might be a fish.
NOAA ship Okeanos Explorer will be coming back and continuing exploration of this
Just
absolutely
back,
stunning place and ecosystems that are found within the sanctuary.
There it is.
Oh, yeah.
I think this is that blind lobster. Remember that little one we saw earlier? This is the adult form. It's called Homerian asper. I saw the color and I thought it might be a chonax or something.
Or you can push in there.
And you can see its super long arms. So the family it's in is called Polychelidae. Has these really, really long arms that are tucked into the sides of its body. But this animal does not have eyes. Thanks, Jake.
Okay.